I want you to just take a second and imagine your backyard.
Or even, the massive farms that are growing the food sitting in your fridge right now.
Right, exactly. Usually when we think about fighting off pests in those spaces, like
Hmm.
mosquitoes, ticks, crop-eating beetles, we picture this, like giant chemical sledgehammer.
Yeah, you just spray a broad spectrum poison, nuke the entire area, and basically just hope for the best.
Right. But today, our mission for this deep dive is to decode this massive, completely invisible arms race that is, well, retiring that sledgehammer forever.
It's a total paradigm shift.
It really is. We're going to look at everyday consumer behavior, global agricultural guidelines, and some seriously wild laboratory breakthroughs to show you how science is swapping out those old poisons for like genetic scalpels and microscopic predators.
They're highly targeted precision blockers, too.
Yes. So whether you're a homeowner just trying to keep ticks off your rescue dog or you're, wildly curious about genetic engineering, we're going to completely change how you look at a simple can of bug spray.
But to understand that shift, we really have to look at the pressure cooker that's forcing us to abandon our old tactics.
Right. We can't just keep doing what we've been doing.
Exactly. We're dealing with this convergence of three massive forces. You've got extreme environmental shifts, changing consumer demands, and the relentless engine of evolutionary biology.
Let's actually start with the environment because it's something you can literally feel just by stepping outside. We're seeing some of the hottest summers on record, punctuated by these intense, heavy rains.
Yeah, the climate is definitely turning up the heat on this issue.
Totally. And the pest control brand OD tracks these environmental windows. And they're seeing the active seasons for mosquitoes and ticks stretching out way longer than ever before.
Well, yeah, because heat accelerates the life cycle of these insects, and that heavy rain, it provides the perfect breeding grounds. I mean, a mosquito doesn't need a whole swamp to lay eggs.
No, it's, what is it, like a bottle cap?
Literally, an amount of standing water the size of a discarded bottle cap is a totally viable nursery for them.
That's great.
Right. So when you multiply millions of bottle caps, clogged gutters, damp leaf piles across a warming country, the population pressure just becomes exponential.
And at the exact same time the environment is heavily favoring the pests, our tolerance for how we fight them has plummeted. Like during the COVID-19 pandemic, 1 in 5 American families adopted a pet.
Oh, yeah, backyards suddenly became the primary playgrounds for millions of new dogs and cats.
Exactly. So now, nearly a third of all consumers are actively demanding natural, eco-friendly pest control. I mean, nobody wants to unleash a broad-spectrum neurotoxin on the exact patch of grass where their golden retriever is rolling around.
Right, people are realizing that the sledgehammer hits the things we love too. But
Yeah.
the third force is the one that's really keeping scientists awake at night.
The evolutionary stuff.
Yes, Darwinian selection happening in real time. Pests are literally evolving to survive our chemical arsenal.
The Insecticide Resistance Action Committee, or IRAC, they track this globally, right?
They do. And they define resistance as a heritable change, where a previously effective product it simply fails. The bugs survive, they breed, and they pass that chemical immunity right on to their offspring.
The way IRSIC categorizes these chemicals is actually brilliant. They use a mode of action classification. I kind of think of it like a football playbook.
Okay, I like that analogy.
Right. Like if our defense runs the exact same blitz every single time, the opposing quarterback is going to figure it out, adapt, and build a counter strategy.
Oh, no, absolutely.
So if we keep spraying the same class of chemical, the surviving pests build genetic defenses. The playbook basically dictates that we have to rotate our plays to keep the pests guessing.
But rotating the play doesn't just mean, buying a different brand name with the exact same active ingredient. You have to rotate the actual physiological target inside the insect.
So attacking a completely different system.
Exactly. If your last spray targeted the pest's central nervous system, your next application needs to target, say, its midgut or its respiratory muscles. You have to hit a completely different biological pathway.
But the crisis we're facing is that we are kind of running out of new plays, right?
We are. The old chemical playbook is largely obsolete against these highly adapted super pests now.
And because developing a brand new, safe synthetic chemical takes years and billions of dollars, scientists have to look for incredibly specific vulnerabilities. We are moving from carpet bombing to sniper rifles.
Which brings us to a perfect example of this new precision chemistry, a newly developed compound called acinonapure.
What's it out of here?
Yeah, it's a fascinating breakthrough from Nippon Soda Company. In the IRAC playbook, this represents an entirely new mode of action, group 33.
Okay, group 33, what does it do?
It is classified as an acaricide, meaning it's engineered exclusively to kill mites, specifically the spider mite, which is notoriously destructive to crops and incredibly fast at developing resistance.
And looking at the chemical structure, it features an azabicyclic ring and an oxyamine structure,
Look,
which...
alchemistry there is just brilliant. The way those structures interact.
Wait, hold on. Let me jump in and translate that from chemistry jargon for a second. We're basically talking about the literal structural shape of the molecule acting like a physical wedge on a microscopic level, right?
Yes, exactly. It's designed to fit into a very specific slot in the mite's biology.
Okay, so let's look at the cellular mechanics of how that wedge works. Inside the nervous system of a spider mite, there are these microscopic gateways called KCA2 channels.
Right, the calcium-activated potassium channels?
Yeah, and their job is to regulate the flow of potassium ions, which in turn controls the electrical firing of the mite's nervous system. So encinodopyr acts like a microscopic cork. It physically blocks that specific channel.
And if the potassium can't flow, you are basically causing a catastrophic short circuit in the mite's brain. Total neurological chaos. Yeah, by halting the flow of ions, the mite's nervous system experiences this abnormal electrical excitation. They did these electrophysiological patch clamp studies.
But what does that mean?
Oh, it's where scientists literally measure the electrical currents across individual cell membranes.
That is incredibly tiny.
It is, and they showed that even at microscopic concentrations, this blockage causes severe convulsions. The mite becomes completely paralyzed and dies shortly after.
See, what's wild to me is that the discovery of this hyper-specific molecule still involved a massive amount of laboratory trial and error.
Oh, completely?
Like, they didn't just mathematically generate the perfect spider mite killer from scratch. They started with lead compounds structurally similar to other known calcium channel blockers, and then systematically tweaked the recipe, synthesizing and testing variations until, bam, a spider mite has a seizure.
Yeah, modern chemistry is often a process of directed evolution in the lab. You build hundreds of keys until you find the one that turns the lock.
And the crucial advantage of encinodopyr, and why it solves that consumer demand for safety you mentioned, is its unbelievable selectivity. It has virtually zero activity against mammalian calcium channels.
Right, so it doesn't fit the lock in a human, a dog, or a cat. It literally just bounces off.
That is so cool.
It is. And furthermore, it has minimal impact on beneficial insects like honeybees. It is a highly specific weapon aimed entirely at spider mites, leaving the rest of the ecosystem completely untouched.
But, synthesizing a new molecule like encinodopyr is an incredible feat, but it's also incredibly difficult, which led researchers to a completely different realization. Why spend billions inventing a new chemical wedge when nature has spent millions of years building perfect weapons?
Yes, this pulls us right out of the chemistry lab and into the biological boom.
And Brazil provides the perfect case study for this shift. As an agricultural superpower, Brazil is currently experiencing an absolute explosion in the biopesticide market.
We are looking at a compound annual growth rate nearing 50%. The market is rapidly approaching $700 million.
So this isn't just some niche organic farming trend.
No, not at all. This is massive commercial-scale deployment.
And the scale of the threat explains the rapid adoption. Nematodes alone, these microscopic roundworms that attack and destroy plant root systems, they cause an estimated $11.7 billion in agricultural losses in Brazil.
Yeah, $11.7 billion. When traditional chemicals fail against numbers like that, farmers have to unleash living predators.
So they are deploying specific strains of fungi like Trichoderma and Beauveria bassiana, alongside bacteria like Bacillus thuringiensis, which is commonly known as break.
The mechanism of trichoderma is honestly like watching a microscopic horror movie.
Oh, totally.
It engages in mycoparasitism. It actively hunts down plant pathogens in the soil.
I picture this as releasing microscopic wolves into a cornfield. But instead of teeth, these fungal wolves use chemical warfare.
Huh, that's a great way to put it.
When the Trichoderma fungus finds a pathogen, it physically coils around it. Then it secretes enzymes called chitinases. Since the cell walls of many fungi and insects are made of chitin, these chitinases literally melt the structural shell of the enemy, allowing the trichoderma to just absorb its nutrients.
It's brutal, highly effective, and entirely natural. And Brazil's pivot to these biological weapons was supercharged by a crisis back in 2013.
Oh, the caterpillar outbreak.
Yeah, a massive outbreak of a caterpillar known as Helicoverpa armigera swept through their crops. The traditional chemical pesticides farmers had relied on for years, completely failed.
The caterpillars had built up resistance.
Exactly. Farmers were staring down total crop failure, so they were basically forced to adopt biologicals, specifically baculoviruses and Bt, just to halt the devastation.
So the biologicals saved the crops by simply introducing a predator the caterpillars had absolutely no synthetic defense against. But to me, the most elegant part of using a fungus like trichoderma isn't just the assassination of pests, it's how it interacts with the crops themselves. It basically acts as a plant vaccine.
Yes. We call this systemic acquired resistance, or SAR. In the context of integrated pest management, we want to look at the whole ecosystem. When a beneficial fungus colonizes a plant's root system, the plant's natural immune system detects a mild intrusion.
So the plant realizes there's a foreign entity on its roots,
Right.
and it goes into a low-level defensive posture.
Exactly. The entire plant begins to physically change. It thickens its cell walls, making it harder for actual pests to chew through the leaves. It starts stockpiling defensive enzymes and proteins system-wide.
That is amazing.
Right. So by the time a deadly pathogen or a nematode actually attacks, the plant is already fully armored. The biological agent trained the plant to defend itself.
Okay, so we've gone from chemical wedges to microscopic fungal wolves and plant vaccines, but science is pushing the boundary even further. If biologicals are about releasing natural predators, the next phase is hacking the pest's own biology from the inside out.
We are now fighting pests using their own genetic instruction manuals against them.
Which brings us to RNA interference, or RNAi. This is arguably the most monumental leap in pest control technology we've seen in our lifetimes.
Without a doubt. Late in 2023, the EPA approved a product called Leadprona, marketed as Calantha. This established an entirely new IRS playbook category, Group 35.
Leadprona is the world's first sprayable RNAi insecticide.
And to understand the gravity of sprayable RNA, we need to look at what it targets. Leadprona was engineered specifically to eradicate the Colorado potato beetle.
Which is a pest that has developed resistance to over 50 different chemical insecticides, right?
Yeah, it's incredibly stubborn. But inside the cells of that beetle is a gene designated as PSMB5. This gene is vital because it regulates how proteins fold within the cell.
And if proteins don't fold into their correct three-dimensional shapes, they can't do their jobs. They just turn into cellular garbage, the cell functions collapse, and the organism dies.
Exactly. So Leadprona operates by intercepting the instructions to make that specific protein. The insect's DNA is the master blueprint stored in the nucleus. When the cell needs to build the PSMB5 protein, it sends out a copy of the instructions using messenger RNA, or mRNA.
Okay, so I think of the bug's DNA as the factory headquarters. It's sending out blueprints, the mRNA, down to the factory floor to build the survival proteins. The RNAi spray acts like a microscopic mail sensor.
Oh, I like that.
Right. When the beetle eats a potato leaf sprayed with Leadprona, the double-stranded RNA in the spray finds that specific blueprint in the beetle's system and literally shreds it before it ever reaches the factory floor. The factory shuts down, the cellular garbage builds up, and the beetle dies.
The mail sensor analogy captures the mechanism perfectly. It's intercepting the message. It is not altering the DNA headquarters itself.
Which is a really important distinction.
It is vital for understanding the safety profile of RNAi technology.
Because from a listener's perspective, hearing the words genetic spray and RNA immediately triggers alarm bells.
Oh, sure.
If you are spraying a genetic off switch onto a potato field, what happens when the wind blows and it drifts into a neighboring yard? Or what happens when a honeybee lands on the leaf or a humie eats the potato?
Well, the specificity of RNAi is its greatest safety feature. The RNA sequence in Leadprona is an exact microscopic match to the Colorado potato beetle's genetic code. If a honeybee, a bird, or a farm worker ingests it, the spray does absolutely nothing.
Because the matching sequence simply does not exist in our genetics.
Exactly. Going back to your analogy, the mail sensor is looking for a very specific envelope. If it doesn't see that exact name and address, it just lets the mail pass through untouched.
Okay, that answers the biological drift question, but what about environmental accumulation? The old chemical sledgehammers lingered in the soil for years, leaching into groundwater.
Well, because Leadprona is fundamentally just a sequence of genetic material, it degrades naturally and rapidly when exposed to the elements. EPA data confirms that the double-stranded RNA breaks down in soil within half a day to 3 days.
Wow, that fast.
Yeah. It is so hyper-specific and degrades so rapidly that the EPA actually waived their standard toxicity testing for fish and cancer testing for humans. The environmental footprint essentially vanishes in 72 hours.
We are spraying a temporary invisible code that turns off a single gene in a single species of beetle and then dissolves into the dirt over a weekend. It's staggering.
It really is.
But this level of unbelievable precision introduces a new problem. A sledgehammer is easy to use. A genetic scalpel requires exact handling. All of this laboratory brilliance means absolutely nothing if the application on the ground is reckless or incorrect.
Very true. The tools have evolved, which means the instructions for using them must evolve alongside them. This brings us to the logistical reality of modern agriculture and the people who actually handle these products.
Right. And the EPA recognized this gap between high-level science and on-the-ground application. Through the Pesticide Registration Improvement Act of 2022, also known as PRIA 5, they are mandating a massive, sweeping transition to bilingual labeling across all pesticide products in the United States, specifically integrating Spanish translations.
Yeah, and utilizing the EPA's standardized Spanish translation guide, the most critical operational and safety data is being translated. This isn't just a bureaucratic update.
No, it's totally unnecessary.
When you are dealing with complex mode of action rotations to prevent resistance, farm workers need to clearly understand the application rates, the required personal protective equipment, the signal words for toxicity, and the immediate first aid protocols.
And this rollout is operating on a strict, prioritized schedule monitored by an electronic tracking system called MyPest. They are forcing companies to start with the most dangerous chemicals first. Any product classified under acute toxicity category, I must feature fully compliant bilingual labels by December 2025.
And then the entire remaining catalog of pesticides must comply by 2030. Translating the science into actionable, accessible language is really the crucial final step in the technology pipeline.
Because a breakthrough biological spray will fail if a worker, say, mixes it with chlorinated water that kills the living microbes before they ever hit the leaf.
Exactly. Clear communication is a technological safeguard in itself.
And this philosophy of precise application trickles all the way down from the massive commercial farms right into your own backyard. Going back to Oh Deer, their pest control strategies rely heavily on integrated pest management, and their core tenet is establishing action thresholds.
Right, because seeing a single mosquito on your patio does not mean you declare chemical warfare on the entire lawn.
No. IPM demands preventative mechanical action first. Before you reach for any spray, natural or synthetic, you remove the habitats. You eliminate the damp, cluttered brush where ticks hide.
You empty that bottle cap of standing water where mosquitoes breed.
Exactly. You change the environmental conditions so the pest can't thrive in the first place. Applied knowledge is the ultimate pest control. You don't need to silence a beetle's genes if you simply disrupt its habitat.
It's all about working smarter, not harder.
So let's zoom out and look at the massive journey we've mapped out today. The battle against pests has fundamentally transformed because it had to. The old poisons were destroying our ecosystems and driving pests to evolve into untouchable superbugs.
And in response, we replaced broad-spectrum toxicity with staggering molecular precision. We are plugging specific KCA2 ion channels in mites to cause neurological short circuits.
We are deploying predatory fungi equipped with chitin-melting enzymes to hunt pathogens and vaccinate our crops.
And we are utilizing sprayable RNA to intercept cellular blueprints and quietly turn off the vital genes of resistant beetles.
All of this innovation is driven by a profound need to protect our warming climate, keep our newly expanded pet families safe, and secure the global food supply against microscopic threats that adapt with terrifying speed.
We've completely changed the weapons, shifting from brute force chemistry to harnessing the fundamental building blocks of biology and genetics.
The sledgehammer is gone, and the scalpel is getting sharper every single day. But to leave you with a final thought to chew on as we wrap up this deep dive, this arms race is far from over.
Not even close.
Today, scientists are using artificial intelligence to bioprospect, scanning the globe's data for new killer microbes and novel RNA sequences. But biology always pushes back.
Yeah, I mean, if we are relying on sprayable RNA to silence pest genes, the inevitable evolutionary response is already brewing. Will these beetles eventually evolve specialized enzymes, molecular scissors of their own, designed specifically to chop up our RNA eye sprays before they can ever reach the cellular factory?
That is, wow. Have we actually ended the war, or have we just shrunk the battlefield down to the invisible genomic level where the counterattacks happen inside the DNA itself?
Really makes you wonder.
It does. The next time you walk out into your backyard and spot a tiny bug resting on a leaf, we hope you look at that incredibly complex microscopic conflict in a completely new light. Thank you for joining us on this deep dive.